Okay, so a very good morning to all of you, and we are going to continue our discussion today on levels of organization that we started yesterday. And we spoke about whole organism as one of the levels of study of metabolism. So today we will be moving ahead to understand how an isolated organ could be a level of organization, and we will also see what is the concept of perfusion. Okay, there is a very popular concept called as organ perfusion. So we will see what do they actually do to a isolated organ, how do they study metabolism in an isolated organ. Okay, so that's something that is going to be the focus today in the first 5 to 10 minutes. So, to begin with, we need to understand that at many instances, studying an isolated organ, or rather studying a whole organism, would be difficult. Okay, and that's the reason why scientists prefer studying isolated organs and tissues. Okay, because you know that in whole organism there might be difficulties related to transporting materials like the radio isotopes that you make use of to a desired site. So you have to inject it into the bloodstream, but then it's not under your control after injecting it into the bloodstream whether it's being transported to the required site or not. So these kind of issues could be, avoided using an isolated organ. Okay, so basically after an organ is removed, the researcher is going to perfuse it. Okay, now this means that perfusion means that you are going to pump specific solutions into the organ and out of the organ. So just like circulation would have taken place inside a whole organism, similarly you are here going to expose the isolated organ to a specific solution. Okay, so this movement of solution in and out of the organ is what we call as perfusion. Now, for perfusion, researchers normally pump a buffered isotonic solution, okay, containing whatever is your study material. That could be some nutrient, radio-labeled materials, or it could be drugs if you are studying the effect of drugs, or even for that matter, hormones could be used. Okay, so whatever is your study, accordingly, the material could be added to the buffered solution, and that could be in turn perfused into the organ. Okay, so now we need to understand that just like circulation, this material will be delivered to the organs. Okay, and whatever is the waste product that's being generated is going to be, coming out of the perfusion fluid, okay, via the perfusion fluid. Now, what you need to understand is that many a times, if this would have to be done in an whole organism or in an animal, surgical procedures would have been involved, invasive techniques would have been involved, and there are a lot of ethics associated to it. So why not then study it in an isolated organ rather than studying it in whole organism? And most of the times these organs, come from a very recently dead animal. So they remove the organ before the organ is totally gone and out of use. And then they study the effect of whatever materials they want to, or whatever metabolic profiling they want to on this isolated organ using the perfusion method. But yes, even though we say that this has advantages, every method has its own challenges. It has its own disadvantages. So kitna bhi tum perfusion kar lo, it's not going to be a perfect match of circulation. It's not going to exactly mimic the process of circulation. So that is one of the major disadvantages that you will see when you talk about perfusion. Okay, so secondly, you need to understand that this cannot be forever, because you can do this till the time the cells in that organ are intact and are alive, because ultimately you have isolated that organ out of the living system. So it's not going to be a forever model system. It's going to be only for a limited duration. So you need to understand that till the time the cells are there, alive, working, intact inside the organ, your study should be complete. Right? Now, many a times, to just mimic the efficacy of circulation, what people do is that they introduce cuts in the tissues, okay, such that thin slices are formed, and then they begin with the experiment. Now, why is it so? This is because so that the materials that are being transported via perfusion, they can travel into nook and corners of the organ. See, in case of circulation, because the blood vessels are so properly, oriented over the organ and into the organ, it ensures that the materials are being transported to every nook and corner of the organ. But that may not be the same with perfusion. So to bring the same kind of efficacy with perfusion, you will have to introduce some cuts into the organ and see to it that the perfusion fluid is bathing every nook and corner of that organ. Now, surely this is going to affect the structural integrity of the organ, right? But most of the cells are intact. So if you are studying something related to what happens at the cellular level, what happens at the molecular level, I think this is really very much fine, because we are nothing to do here with the anatomy of the organ. We want to try to understand the physiology of the organ. So even if the structural integrity is affected, that is still acceptable. Okay, only thing that you need to see to it that you want to keep the cells alive till the time your study is going on. So you keep it oxygenated, provide it enough oxygen, and provide it the substrates that it would need to survive. So it should be better oxygenated, it should be provided with substrates so that the cells are alive. Now let me tell you that the Krebs cycle that you study, the citric acid cycle or TCA cycle as you call it, the entire pathway has been elucidated by doing experiments with slices of liver and heart. Okay, so it was like our organ perfusion method that was carried out by Hans Krebs, Sir Hans Krebs, who is the person who has elucidated the Krebs cycle for us, right? So it was using this technique of organ perfusion. So on the slide, you can right now see how they have a heart which is being maintained in a chamber to maintain similar kind of conditions, and it is being perfused with material into it, right? And it's just like a very mechanical process. That's why I've shown you that GIF on the slide right now, that it's a highly mechanical process, and it's very difficult to mimic the entire system as it is, but then we are trying to do that in this particular mechanism. Okay, but then as I said that there are going to be its own set of challenges. You should make choice of this method only if you feel that this method is going to help you in elucidating the metabolic profile that you are trying to study, because this may not be a suitable method for all kinds of metabolic activities that are studied out by researchers. Okay, so you will have to decide on the basis of what you are studying that whether this method is really going to be beneficial for you or not. Okay, so this is about isolated organ. Most isolated organ studies have been done with heart, liver, pancreas, even kidneys to a certain extent. So moving ahead, let's go to the next part, which is whole cells. So we need to understand that, a whole cell, that is, we are not talking about a tissue or an organ. We are speaking about disintegrating a tissue into its basic components, which are the cells, right? So that's the GIF showing out there, that you are not studying the organism as a whole, but you are trying to disrupt the constituents of the organisms into single entities, which we refer to as cells, right? So we need to understand that cell, which we refer to as the basic structural and functional unit of life, is one of the best models that biochemists utilize to study metabolic pathways. Okay, so you might be knowing that there are materials like trypsin, enzymes like trypsin, collagenase. These are those enzymes which are used to disrupt the tissues into individual cells. Okay, so you'll be able to use these and disintegrate the tissues to form single cells. So in any plant tissue culture or animal tissue culture setup, you will see that this is one of the most common steps wherein they are disintegrating the tissue to form individual cells. Okay, so what does this enzyme basically do? It's going to break, it's going to act on that extracellular matrix. It's going to act on those proteins, on those components, which is holding the cells together to form that layer of cells which we call as tissue. Okay, so अगर उस adhesive को ही हमने तोड़ दिया जो cells को साथ में पकड़ के रख रहा है, then you are getting your individual cells. Now, for plant tissues, you may have different set of enzymes because trypsin and collagenase is normally something which is used for animal tissues. But for plant tissues, you will see that they will use enzymes like pectinases, cellulases to disintegrate the cells from the tissues. Okay, so you need to remember this because there the cell walls of the plant cells are responsible for holding the cells together. So something acting on the cell wall should be enough to disintegrate the plant cells from their tissues. Now we need to understand that any organ of an animal or any part of a plant, it's going to not have a single type of a cell. Okay, it's always composed of different types of cells. Okay, so after disintegrating, you can't say that you have a homogeneous mixture of cells, which means that the mixture of cells, or rather the cell suspension that you have, it's not going to have a single type of cell. It's going to have variety of different cells present there. And you may not want to study all of them. You may want to study a single type of cell from that mixture. So then how do you go about it? Because now you will have to separate them from the mixture, right? So for this, these days we have high-end instrumentation available. So I don't know how many of you have heard of an instrument called as flow cytometer. So flow cytometer is basically responsible for counting the cells. It was brought in to study or count the eukaryotic cells first, but these days even flow cytometry is being involved in analyzing the prokaryotic cells. Okay, so it is basically wherein the cells are aligned in a single path, and then it is going to come out of an orifice, and these cells are basically tagged with fluorescent antibodies. Okay, fluorescently tagged antibodies. So it goes on counting the number of cells because it will just go through a cell orifice where there is a laser which is going to put incident light on the cells, and obviously if it is having a fluorochrome, it's going to give out fluorescence, and that fluorescence is measured. So every time a fluorescence comes out, it's going to count it as 1, and second time second. So that's how it counts the cells. And this flow cytometry was advanced to make a technique called as fluorescence associated or fluorescence activated cell sorter. Okay, so it's called as FACS, fluorescence activated cell sorter. Okay, so this technique, what it does is, if you have a mixture of cells, okay, please understand that हर cell के पास उसका एक specific surface antigen होता है. जैसे हम वायरस के बारे में बोलते हैं, उसके ऊपर एक उसका यूनिक सरफेस एंटीजन होता है, वैसे हमारे बॉडी के या प्लांट के सेल्स के सरफेस पर you will find specific antigens which is very unique identity of that cell. वो आपको उसी ऑर्गेनिज्म के किसी और सेल पर दिखाई नहीं देगा. ओके, सो इफ यू नो कि आपको जिस सेल में इंटरेस्ट है, जिसके बारे में आपको पढ़ना है, उसके सरफेस पे कौन सा एंटीजन है, तो आप उसके खिलाफ जो एंटीबॉडी है उसको फ्लोरोक्रोम के साथ टैग कर सकते हो, and you can add it to the cell mixture. So what would happen? Antibody will specifically go and bind to the cell type that you want to separate and study. Okay, अगर आपको 2 या 3 टाइप्स में इंटरेस्ट है, और लेट अस से दैट हमारे सस्पेंशन में 6 सेल टाइप्स है और आपको उसमें से 3 पढ़ना है, और अगर आप उन तीनों के बारे में उनके कौन से एंटीजेंस है ये जानते हो, तो उनके स्पेसिफिक एंटीबॉडीज का इस्तेमाल करो, उनको अलग अलग फ्लोरोक्रोम्स के साथ टैग करो. तो अगर उन सेल्स को मैं ABC नाम दूं, तो A के और एंटीबॉडी के लिए जो फ्लोरोक्रोम होगा वो रेड कलर का होगा, B के लिए जो एंटीबॉडी होगा उसका फ्लोरोक्रोम ग्रीन कलर का होगा, और C के लिए जो फ्लोरोक्रोम होगा उसका येलो कलर होगा. So I have given it, I have tagged the antibodies with different fluorochromes, okay, different colored fluorochromes. So what happens is when the cells are aligned, okay, and when it is coming out of the orifice, it's going to be excited by the laser. Fluorescence is going to be given out. That fluorescence is going to be recorded, And depending on the color, okay, it's going to be sorted. Now, this sorting happens on the basis of charge because these cells will also be given a charge. So I will show you the GIF. I'll go back to the slide. So you can see here what's happening. Can you all see working? Is the GIF working in front of you? Are you able to see it? Can someone say a yes or no?
Yes, sir.
Yes, sir.
Yes, sir.
Great. So can you see that first the sample is loaded at the top, okay? The cells are being aligned in a single order. So remember that through the orifice is decided in such a way that only one cell can come out of it at a time, okay? So you can see that as it comes out, the laser beam is incident on it. You can see that the color is being detected, and on the basis of that, can you see that all the cell types are being sorted? So this is one of the best ways to sort the cells. But let me tell you that this instrument is extremely expensive. Okay, and FACS is really very expensive, and its maintenance is also equally expensive. So, you know, very few institutions have it. We have FACS in NIRRH, which is the Reproductive Research Center in Mumbai. Okay, we may also have it in IIT. I'm sure they would be having it. But one of the most famous FACS centers in Maharashtra is NCCS, okay, which is the basically National Center for Cell Sciences. So they obviously, as the name suggests, are into cell biology. So they have a dedicated FACS facility wherein they have around, if I'm not wrong, they have somewhere between 7 to 11 FACS instruments, and they train researchers to use FACS, and they also allow such trained individuals to come and analyze their samples on their own. So if you have FACS work in your project, in your research, they will first make you go through a training process, post which you have access to use those instruments, obviously for a payment, because otherwise they will not be able to maintain the facility. And you can book the slots, okay? Their booking slots are always occupied, and it's a 24/7 facility. So, you know, even people go there in the midnight and run FACS because that's the demand for it, because every institution may not be able to afford it, okay? Fine. So what I have done is, the entire principle is not in the scope of our study of these instruments. So I have posted 3 videos, video for NMR, video for flow cytometry, and video for FACS on the website. Okay? 2 of them, the flow cytometry and FACS ones, are iBiology videos, and NMR is a Bruker Corporation video. So you can go through all of them. iBiology videos may be, you know, huge. There's a lot of information in it. But I would say that whenever you get time, you know, you dedicate 5 minutes, 10 minutes on a daily basis, and then continue the next day from that point. That still helps, because the more information you gather, you may not be able to understand everything, that's fine. But the more you know about things, it becomes easy for you to link things when you are asked questions about in interviews, or you're writing your SOPs for your master's admission. So it's always better to know more. Okay, there's no harm in trying to understand new concepts. So it's up to you. I have put up the links on the website. You can play the videos there itself. You don't even have to toggle. So it's completely up to you to go through them. Fine. Now let us continue with our discussion of whole cells. So when you talk about sorting the cells, you know, especially this is of huge help in cancers, because when you have you know, sample coming from a cancer patient, it may have both normal cells and cancerous cells, right? And cancer cells normally have specific marker antigens on their surface. So if I want to study just those, I would want to separate them from the normal cells of the patient. So FACS is the best option for that, right? So you will see a lot of cancer research institutions having this. So yes, ACTRAC also has an FACS. Okay, so it's very important for them, because it's on a daily basis that they need to sort the cells and then subject them to specific studies, right? So FACS advantage is that it could sort around 1,000 cells per second. So can you imagine? The speed is phenomenally great, that it's sorting around 1,000 cells per second. So it's amazing. And even with a small sample size, you still get a very appropriate segregation of cells, which is very good for researchers. Now, if you know you want to study these cells further, you may have to go for tissue culture. Okay, because the samples coming from patients is not going to have a huge number of cells of your interest. So you may have to cultivate them. But it's very important to understand that when you're trying to cultivate, especially human cells or plant cells, you need to know what their specific requirements are. Okay, if you don't provide them those growth factors, if you don't provide them those nutrients, they may just not grow. They are not like bacteria. Okay, bacteria is still comparatively easy to cultivate as compared to mammalian cells or plant cells, because they are comparatively simple in their requirements. I'm not saying that all bacteria are so, but as compared to mammalian cells, their requirements are quite simple. Okay, so you need to provide them the nutrients and the growth factors that they need, and only then they are going to divide and grow, so that you get a large number of these cells for your study, right? But you need to understand that with animal cells, there is another complication. The complication is that animal cells cannot divide forever out of the system. So after few rounds of multiplication, they will stop dividing. Okay, they won't multiply post that. So in those circumstances, you need to understand that variants of these cells, which have gone cancerous, they can multiply continuously. Please understand this. A normal animal cell is not going to multiply forever. But if that cell line is made to go cancerous, if that cell is made to go cancerous, then it's going to multiply like forever. Okay, that's what we call as cell lines, which are normally used, in the pharma companies, in the cosmetic industries, to check for effects of drugs. Instead of directly using it on animals, you use a cell line of liver, you use a cell line of kidney, and you see what happens when you expose it to this drug, which you want to release it in the market. And if you see that the cell is getting affected, if the cell is dying, or it is showing some kind of toxicity-based symptoms, then it's obvious that, okay, your drug is risky. So you are not trying it on animal, you are saving the animals. So the ethics aspect is handled, and you are checking still the efficacy on the cell lines, which gives you a reliable data. Similarly for cosmetics, if you want to test if the product is safe enough and is not inducing any kind of allergic reactions, then cell lines could be used, right? But we really need to understand that these cell lines, especially the cancerous ones, they go on losing their properties over time. तो जैसे बैक्टीरिया में हम बोलते हैं कि जितना तुम सबकल्चर करोगे उतना उसका प्रॉपर्टीज इफेक्ट होता है, उसी तरीके से मैमेलियन सेल्स में भी जितना आप राउंड ऑफ मल्टीप्लिकेशन करते जाओगे, you will see that its characteristics go on changing as compared to what the original cell was, which was not even carcinogenic, which was not not carcinogenic, not cancerous. Okay, so its properties are lost eventually. So then that is something very difficult to deal with, because then your study is not going to yield reliable results for you. So such cells which come from a single cell are called as clonal cell lines. Why clonal? Because they are expected to be clones of that single cell. Okay, especially for first few rounds of division, they are going to maintain the characteristic features of that parent cell. So we call them as clonal cell lines because they are clones, they are copies. Okay. Now, this system is also utilized to study viruses, because I don't want viruses to be introduced into an animal or into a human and then study it. I'll be in the jail. Okay. So we can introduce the viruses into these cells. So, you should know that what's the target cell for the virus. And accordingly, you have to have that cell line and introduce the virus in study. Even for vaccine developments against the virus, these cell lines are used to a very, very great extent, right? Other than this, what you need to understand that the challenge here, as I've already mentioned, is that number one, maintaining these cell lines is very difficult. Okay, because for long-term use, you really have to struggle hard to maintain these cell lines. So such cell line collection banks, they have a huge task at their hand. Okay, and it's an expensive process as well, because the nutrients that they need are really very expensive. Okay, serum is used at times as nutrients, so you can imagine that's going to be really very expensive. Other than that, the property of laws of characteristics over time as compared to the parent cell is another challenge that needs to be dealt with, right? So these are the two things that you need to keep in mind when you talk about whole cells. So this is just to indicate the tissue culture mechanism. Now we go to cell-free systems. So cell-free systems is where we are talking about breaking the cells open and trying to analyze them and trying to study the metabolic characteristics, right? So this may not be a very preferred mechanism for all types of studies, but it has been noticed that it is something very beneficial to study compartments of the cells, like specifically studying mitochondria, or studying chloroplast, or studying endoplasmic reticulum, Golgi apparatus. So for those purposes, this may be a best mechanism, right? So we need to understand that when you break open the cell, for breaking open the cell, there are different methods. You have studied this in SY as well, that you could use physical methods, you could use chemical methods. But whatever method you are going to make use of, you need to also remember that that method may have an effect on the organelles that you are trying to separate and study. Because the membranes of the organelles have a similar composition as compared to the cell membrane. So whatever agent you are making use of, that could actually affect the integrity of the organelle also that you are trying to study, right? So that has to be always kept in mind. So can you tell me what are the basic methods that you have learned of in SY for cell lysis? Yes, can I hear from you guys? You can also type it up. That's completely fine. Homogenization.
Sonication.
Sonication. Then
Freeze thaw.
freeze thaw. Okay.
Grinding, normally breaking the cells.
Grinding the cells, that is having shear forces, right? Even simple, techniques like, I don't know if you have heard about this, you would have heard that just passing the cell suspension through a pipette multiple times leads to lysis of cells. Okay, because that's also a shear force. So if the pipette orifice is comparatively small, pipetting it multiple times also leads to a shear. Okay. Also adding the cells to hypotonic solution, right? That could also be used for lysis of the cells, barabar? Or you can also make use of enzymes like lysozyme, which is going to act on the bacterial cell membrane and break it open. So if you're making, a bacterial cell lysozyme is a good option, right? Other than that, if you're making use of yeast or plant cells which have a comparatively tough cell wall, then you may have to use a combination of enzymes like cellulases, hemicellulases, pectinases, which will act on individual components of the cell wall and break it. Mechanical treatments, like someone very correctly said, that grinding it or making use of shear forces. You would have even heard of instruments like U-press, suna hoga na, SY mein, which could be utilized to break the cells open, right? So you need to understand that once you break the cells open, all the kachra of the cell is going to come out. Okay, so that's the cell-free homogeneity. It's going to have all the kachra that's there inside the cell. Now, if you want to study a particular metabolic process which takes place inside a specific compartment, inside a specific organelle, then your task begins here. You will have to start fractionation. What is fractionation? Whatever homogenate you got, you want to separate it into different fractions so that every fraction is going to have a specific component or specific organelle. So for this, what will you do? You have studied this technique in SY practicals. What will you do? You will take a sample of the cell, right? You will take a sample of the cell, you will what will you do to separate components?
Gradient centrifugation.
Very good. So you will do differential centrifugation, wherein you can go for density gradient centrifugation. So if you're separating biomolecules, it's very much possible doing that with density gradient centrifugation, right? So you could use sucrose gradient, or you could use gradients made up of ficol hypake, which you can use to separate biomolecules. Similarly, you can separate organelles also, because organelles also are of different intensity. So you can separate it at different speeds, at different time lengths, and accordingly, nuclei, mitochondria, chloroplast, Golgi apparatus, lysosomes, and endoplasmic reticulum. You don't get the entire endoplasmic reticulum ever, because when you break the cell open, endoplasmic reticulum being very delicate, even the endoplasmic reticulum breaks. But जो fragments बनते हैं endoplasmic reticulum के, they circularize themselves and reseal themselves. Okay, so the fragments of those endoplasmic reticulums which have resealed themselves are what we call as microsomes. Okay, what do we call them? Microsomes. So when you are trying to fractionate the cell components and the cell organelles, remember that you will not say endoplasmic reticulum, you will refer to it as microsomes. So these are nothing but artifactual membrane vesicles. Artifactual means something which has been damaged, and it just reseals itself to form circular vesicles. So they are disrupted endoplasmic reticulum, which has just tried to integrate itself on its own after a disruption has taken place, right? So you can separate the organelles. You can also separate the components of the cytoplasm, the biomolecules as it is, the enzymes, the proteins, the other proteins, the lipids, the carbohydrates could be very well separated, right? Most of our understanding of nucleic acids, even specifically DNA transcription, DNA, the process of transcription from DNA, the replication process, all this has come out of isolated nuclei. So nuclei which was separated, and then the process in it was studied. So most of the information of DNA related processes has come from this. Okay. Respiratory metabolism takes place inside the mitochondria has come from the isolated mitochondrial studies. So especially ETC, electron transport chain based studies, it has always occurred in an isolated mitochondrial setup. Now we go ahead to the next part of it, which is the purified components. So after breaking the cell open, we are trying to purify the components from it. And after purifying the components, we are going to reconstitute it. So it's like कुछ तोड़ना और फिर उसको दोबारा जोड़ना. Okay, that's reconstitution. So you first break the cell open, let all the kachra come out, purify the components that you need, and you try to make a setup out of it. To study a metabolic process. That with the purified components, does this system actually work? Okay, so to study something in its intricate details at the molecular level, it's very important that the components are purified. Okay, so even when you talk about TCA, let me tell you that efforts were taken to purify the enzymes involved in TCA, and to show that when you get the substrates and when you get these purified enzymes together, you will still see the reaction taking place. So the entire process could be reconstituted with its purified components in the lab. That's reconstitution, right? But you need to understand that some processes, they're very complicated. They may need a lot of components, right? Like when you talk about protein synthesis, it needs DNA, it needs enzymes for the DNA to be converted to mRNA, you need ribosomes, you need other enzymes. There are so many components involved. So replicating such a setup in the lab is going to be a challenge, because you need all these components ready with you. Only then you can process it out in the lab, right? So that needs to be kept in mind. So purification के वक्त काफी सारे challenges होते हैं, because when you're purifying, there is a huge chance that you lose the component completely if you're not careful. Okay, so that is very, very important. कि तुम ये ध्यान में रखो कि तुमने फैक्टर्स गवा नहीं दिए जो तुमको स्टडी में जरूरी है. ओके, तो बहुत ज्यादा मगजमारी का काम है, बहुत ज्यादा स्ट्रिंजेंट काम करना पड़ेगा so that they are careful of not losing the factors. Okay, many a times purified components का activity थोड़ा कम हो जाता है बिकॉज प्यूरिफिकेशन के लिए तुमने जो भी तरीका इस्तेमाल किया होगा, जो भी केमिकल्स का इस्तेमाल किया होगा, उनका असर प्रोटीन्स पर, एंजाइम्स पर हुआ हो सकता है, जिसकी वजह से एक्टिविटी शायद थोड़ा बहुत कम हो जाए. सो वो याद रखना पड़ेगा. कई बार प्रोटीन्स ना मल्टीपल सब यूनिट से बनते हैं, तो जब तुम प्यूरिफिकेशन करने की कोशिश करते हो तो वो प्रोटीन्स जो असेंबल्ड थे वो डिसअसेंबल हो जाते हैं. और अगर समझो वो प्रोटीन 4 फ्रैगमेंट से बना था, उसमें से 3 ही फ्रैगमेंट प्यूरिफाई होता है, एक फ्रैगमेंट रह जाता है या हम गवा देते हैं, तो वो फंक्शनल प्रोटीन नहीं होगा. तो इन सारी चीजों का हमको खास ध्यान रखना पड़ेगा ताकि प्रोसेस जो हम स्टडी करने की कोशिश कर रहे हैं वो प्रॉपर डिटेल्स में स्टडी हो, वरना वी आर मिसिंग आउट ऑन सम इंट्रिकेट डिटेल्स. So, this is the reason why whole cell, organ level, whole organism level based study is more preferred first, ताकि तुमको एक आइडिया आ जाए कि भाई होता क्या है, फिर बाद में तुम उसको एक्सट्रापोलेट करो टू इन विट्रो स्टडीज दैट मेक सेंस, राइट? So I hope that things still here are clear to all of you. If you have any questions, please let me know, because we have finished the A and B part, which is from Matthew. The reference is already known to you. The book is there on Drive. It's somewhere 500 के range में है pages में, that you will be able to see. First 502 or 3 कुछ तो है. So you will have to check that in the PDF and start reading it. Okay. Any questions that you have till this point, please let me know, or we can move ahead to section C. But before that, if you have any queries, I would prefer solving those first. Anything that you would want to ask?
So can you explain the flow cytometry part again?
Yes. So in flow cytometry, what you do is that there is a sheath fluid. Now this sheath fluid is basically in which you suspend your cells. Okay, and that is going to be responsible for aligning your cells in a ordered fashion. That's one behind the other in a straight line. Okay, and you will see that the passage through which the cells pass, it goes on becoming narrower and narrower. Okay, and it has an orifice at the end. Orifice is an opening. Now the opening is of a size which will allow only a single cell to come out of it at a time, so that when you're counting the cells using a flow cytometer, it is recording for one cell at a time. If the orifice is big and if there is a clump of cells coming out, there might be 10 cells in it and you may record it as 2. We don't want that, right? We want a single cell, every single cell to be recorded for. So the orifice is maintained of a size such that a single cell can come out of it. Now for this cell to be counted, I need a mechanism. So what's that mechanism? We make use of fluorescence. So what do we do is, before the cells are added to the instrument, they are going to be, made to react with antibodies. Now antibodies are going to be highly specific to the antigens that are present on the cell surface. And these antibodies are tagged with fluorescent molecules, which we call as fluorochromes. So these are those tags which, when excited with laser, they give out fluorescence. Okay, so let us say I have 3 types of cells in the mixture. I have used 3 different antibodies targeting, the specific cells, and every antibody has been tagged with a different colored fluorochrome. Okay, so now when I put them into the flow cytometer and it comes out of the orifice, the first thing that's going to happen is the laser light is going to fall on it. Okay, so the fluorochrome gets excited and it gives out a fluorescence of that color. Okay, now this fluorescence is recorded by the detector. So every time a fluorescence flashes out, it's going to record that as one cell. Next one, second cell. Third one, third cell. Okay, and it will be able to record that which colored flashes happened how many times. So that tells you that A ke kitne cell hai, B ke kitne cell hai, C ke kitne cell hai. Okay, and one step ahead is if you want to separate them, then you need to have an electric field based separator, which is FACS, fluorescence activated cell sorter, wherein the cells will be charged as they pass through the orifice, and on the basis of their color they will be separated into different containers. So that is an extension of flow cytometry, which we call as FACS. Flow cytometry basically was just to identify whether I have cancerous cells in this and count them. That's it. If you want to separate them, you need to have an extended ability of that, which is FACS. These days, flow cytometry are equipped with fluorescence microscope also. So you can also observe the cells as they are being, counted or separated. Okay, I hope that answers your question, Sahib.
Yes, sir. Thank you.
Any other questions? Okay, so I don't think we will be able to do much, but I don't want to waste time. We will start with metabolic probes because that's the next topic which we have. So what do metabolic probes speak about? Metabolic probes say that you make use of chemicals which interfere with your pathway. So for example, I make use of inhibitors. वो बोलते हैं ना कि जब कोई चीज नहीं होती है तो हमको समझ में आता है कि इसका रोल क्या है. Okay, so you can make use of inhibitors, you can create mutants, you can introduce mutations and see what happens. Because you know that many a times when mutations disable enzymes, inactivate them, that reaction will not be carried out. So you interfere with the process and try to understand that what's happening in this pathway, what is the significance of this enzyme, what's the significance of this compound. So you try to interfere with the process. That gives you a lot of idea of the functioning. Because when that function or when that component is absent, that's when you realize what it exactly does. Right? So that is very significant. When you block a reaction and you try to understand what is the consequence of it inside that cell. You make use of carbon monoxide, you make use of cyanide. These are all agents which are going to block, which are going to create blocks in biochemical pathways. Like I'm sure that cyanide के बारे में 12th standard में तुमने पढ़ा होगा कि electron transport chain में जो cytochrome C oxidase complex होता है उसमें cyanide block create करता है, because of which ETC doesn't proceed, ATP synthesis doesn't take place, and as a consequence जो भी processes है body में जहाँ पर ATP की जरूरत होती है वो सारे processes बंद हो जाते हैं. Okay, including exchange of ions. So osmotic balance also gets affected, the person dies. Similarly, you talk about carbon monoxide, you should be knowing that carbon monoxide has a very, very high affinity for hemoglobin. So it goes and combines with hemoglobin and forms a complex called as carboxyhemoglobin. अभी कार्बन डाइऑक्साइड या ऑक्सीजन जब हीमोग्लोबिन के साथ कंबाइन करता है तो बेनिफिट ये है कि वो जितने प्यार से एक दूसरे के साथ जुड़ते हैं उतने आसानी से वो अलग भी होते हैं. बट कार्बन मोनोऑक्साइड के केस में एफिनिटी इतना स्ट्रांग है हीमोग्लोबिन के लिए कि वो उसको छोड़ता ही नहीं है, कि वो बोलता है नहीं जाने का नहीं, तू इधर ही रहे. Okay, so what happens is that hemoglobin is now occupied, it's not able to carry oxygen because carbon monoxide is stuck there. Okay, carbon monoxide could also block ETC. So when you use these kind of inhibitors, that's when you understand that okay, what this thing that you're trying to block does actually, what is its function. No doubt that there are disadvantages here as well, that the inhibitor should be properly transported to the site of action. Okay, many a times the inhibitor could have multiple sites of action and then it leads to so many different consequences that you don't understand, because that inhibitor may be blocking ETC also, that inhibitor may be blocking some other pathway also, that inhibitor may be blocking yet third pathway also. So then it becomes difficult कि भाई हम जो पढ़ना चाहते थे उसके वजह से उसको ब्लॉक करने की वजह से कौन सा कॉन्सिक्वेंस आ रहा है, क्योंकि मल्टीपल कॉन्सिक्वेंसेस हैं, क्योंकि ये इनहिबिटर अलग अलग चीजों पर काम करता है. ओके, कंपैरेटिवली कम कॉम्प्लिकेटेड होते हैं म्यूटेंट्स, क्योंकि तुम म्यूटेंट्स वो स्पेसिफिक एंजाइम में म्यूटेशन तुम वो स्पेसिफिक एंजाइम में क्रिएट कर रहे हो, तो तुमको वो टेंशन नहीं होता कि भाई और कहीं से तो कॉन्सिक्वेंस नहीं आ रहा. सो म्यूटेशन बिकम्स अ easier way to handle as compared to making use of these inhibitor chemicals, right? So people have made use of this. You would have heard of Beadle and Tatum. Can someone tell me where you have heard about Beadle and Tatum? Come on. 12th standard mein.
Yes, sir. They formulated the one enzyme hypothesis.
So they were responsible for formulating one gene, one enzyme hypothesis. That एक gene है तो वो एक enzyme बनाएगा. But आज हमको पता है कि ये सच नहीं है. Okay? Because कई बार एक enzyme multiple gene से भी बनते हैं. One gene, one enzyme hypothesis is not really something which is acceptable today. But these are the two scientists who had put up the foundation for that. Okay, the study model was Neurospora crassa. They introduced mutants, or rather mutations in Neurospora crassa by exposing them to X-rays. Okay, and the mutation that they created was that the enzymes which were required for biosynthesis of araginine, which is an amino acid, they were mutated. So the genes coding for those enzymes which are required in biosynthesis of araginine were mutated. Okay, and let me tell you, it's not one enzyme, it's a pathway. Biosynthesis of araginine is a pathway. It involves several enzymes. Okay, so what they did was, let us assume, okay, I'm not saying surely, let us assume that we have 5 enzymes for biosynthesis of araginine. Okay, 5 अलग-अलग enzymes है पूरे pathway में. तो एक Neurospora crassa is mutant in the first enzyme. Second one is mutant in the second enzyme. Third one is mutant in the third enzyme. Fourth Neurospora crassa is mutant in the fourth enzyme, and next one in the fifth enzyme. Okay, so they created mutations in all enzymes of araginine, but in different cells of Neurospora crassa, not in the same one. Okay, so one cell is going to be mutant only for one enzyme. Another one is mutant for yet another enzyme, and so on. So none of these 5 will be able to grow until and unless you provide them araginine. Are you understanding this? So you have made them auxotrophs, the word that Nitin sir used yesterday. You will not be able to cultivate these cells until and unless you provide them this nutrient, because they have lost the ability to synthesize this nutrient on their own. That's what we mean by an auxotroph. That they have lost their ability as compared to their wild type, which has the ability to synthesize it, but they have lost this ability now. So you will have to provide this araginine in the media in which you try to cultivate them, or else they won't grow because they don't have an enzyme. एक भी enzyme अगर अटक गया समझो, वो फिर पहला हो या 5वां enzyme हो, araginine तो बनेगा नहीं ना. So तुमको वो provide करना ही पड़ेगा. यहाँ तक समझ में आया सबको उन्होंने क्या करने का कोशिश किया है?
Yes, sir.
Yes, sir.
Yes, sir.
अभी इससे पाथवे कैसे पढ़ेंगे वो हम बाद में देखेंगे, क्योंकि ये तो सिर्फ स्टार्ट है कि उन्होंने किया क्या, बट इससे पाथवे कैसे पता चलेगा ये तो हमको आगे चल के देखना है. बट यहां तक समझ में आया कि उन्होंने क्या करने का कोशिश किया, तो भी बहुत बड़ी बात है. ओके, सो वी विल कंटिन्यू दिस डिस्कशन in our next lecture. We are going to meet on Thursday this week after your practicals at 3 p.m. Because the topic that we are going to discuss here now for araginine synthesis and how it was studied is slightly complicated. So I want that to happen in this week. So we will meet on Thursday afternoon, 3 o'clock, after an hour's break after your practicals. And we will have a 1 hour discussion. Okay, so please bear with me. The lecture link will be extended for that day, so you can join the same lecture link at 3 o'clock. Okay, so I will stop here. Please visit the website. The presentation for metabolic probes is also uploaded. It has a lot of topics after metabolic probes also. And also some reading material has been uploaded. Though we have not reached there as yet, you can start going through it. Okay. So thank you so much. If you have any doubt, difficulties, please feel free to get in touch. It's already 9:30. So I'll stop here. If you have questions, you can message me. You can also talk to me on Thursday when we meet at 3 p.m. Thank you so much. See you all in the practicals.
Thank you, sir.
Yes, sir. Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.